Assalamualaikum warahmatullahi wabarakatuh and good day. I am Madam Rose Azino. Today we are going to proceed our lecture with cell reproduction. We can start our lecture now with cell reproduction. For the learning outcomes, at the end of the lecture, students should be able to explain factors that cause cells to reproduce, to define cell cycle and explain where mitosis fit into cell cycle, to describe each phase of mitosis, to discuss problems in cell division when control is lost, to distinguish asexual and sexual types of reproduction, to explain the effect that meiosis has on chromosome number, to describe the event of each meiotic phase, and lastly, to differentiate mitosis and meiosis. There are two basic varieties of cell. First is body cell, second is the sex cell. Body cell can be reproduced by the mitosis, which also can be differentiated into special cell types. Arrangement of the elements in the cell varies so that they can carry out various jobs such as digestion, energy storage, or oxygen transport to your tissue. For the sex cells, it is specialized cells that are used to reproduce, and sexual reproduction combines genetic material from two organisms and requires meiosis process. What is the reproduction? Parents produce a new generation of cell or multi-cell individual like themselves. Parents must provide daughter cells with hereditary instruction encoded in DNA and enough metabolic machinery to start up their own operation. For division mechanism, for eukaryotic organism, it can happen in a mitosis and meiosis process, while the prokaryotic organism, it only involved in a prokaryotic fission. Now we look at the roles of the mitosis. Mitosis important in a multi-cell organism, especially in a growth and also cell replacement. For some protistans and fungi, plant and animals, is also function in a asexual reproduction. For the process of the meiosis, it functions only in a sexual reproduction, and it precedes a formation of gametes like a sperm and egg or spores. For cell reproduction, chromosome is very important element. Chromosome consists of a DNA molecule and attached protein. It forms in a duplicate in a preparation for the mitosis. This is the structure of the one chromosome. This is a structure of the one chromosome but in a duplicated form, which consists of two sister chromatids. What is sister chromatids? Each chromosome and its copy stay attached to each other as sister chromatid until late in a nuclear division process. Sister chromatid attach at the centromere. Another important element is the nucleosome. The nucleosome consists of part of a DNA molecule looped twice around a core of histone protein. This is the figure of the organization of the chromosome. Here in this figure, we can see that DNA and proteins arranged as a cylindrical cylindrical fiber. This is the nucleosome, and this is the DNA. This is the histone protein. Chromosome number is the sum total of chromosomes in a cell. Somatic cell consists a number of chromosomes is in diploid, which 2 of each type of chromosome. While the gamete, the chromosome number is haploid, which is N, and also consists 1 of each chromosome types. Basically, for human chromosome number consists of N number is the 46, which is there are 2 sets of 23 chromosomes, 1 set from the father and 1 set from the mother. So the mitosis produce cell with the 46 chromosome, which consist of 2 of each type. Now we move on to the cell cycle. Cell cycle is very important element in the reproduction. Cell cycle start when the new cell forms. During cycle, cell increases in mass and duplicates its chromosome. Cell cycle ends when the new cell divides. Interphase is one of the phase in a cell cycle, usually longest part of the cycle. Cell increases in mass, number of cytoplasmic component double, and DNA is in a duplicated form. Here is the stages in the interphase. Interphase consists of G1 phase, S phase, and also G2 phase. G1 phase is the interval of cell growth before the DNA replicated, which a chromosome is unduplicated form. Next phase is the S phase, where the interval of cell growth when DNA replication is completed. In this stage, the chromosome is in the duplicated form. Next phase is the G2 phase, where the interval following DNA replication, where the cell prepare to divide. Control of the cells. The cell has a built-in molecular brake in G1, what we call as a G1 checkpoint. Once the S begins, the cycle automatically runs through the G2, where the G2 checkpoints and also the mitosis checkpoint. Cancer involves a loss of control over the cycle where the malfunction of the brakes. Here is the diagram of the control of the cell cycle. At interphase, there are G1 checkpoint. If there's something wrong with the cell division, it will go to the G0 stage, which is a resting state. This means the cell will stop dividing. Next phase is the G2 checkpoint. This checkpoint just before the cell start to divide. Next checkpoint is the M checkpoint at the mitosis stage. Some cells stop the cell cycle due to certain circumstances. Some cells normally stop in the interphase, like a neuron in a human brain, arrested cells which do not divide anymore. The adverse condition that can stop the cell cycle, such as the nutrient deprived in amoeba, which can get stuck in the interphase. Do you know about the apoptosis? Apoptosis is defined as a programmed cell death because cell progress through a typical series of events that bring about its destruction. It prevents the tumors from developing. Apoptosis also occurs all the time, particularly in an abnormal cell could become cancerous. How the apoptosis happen? Apoptosis process happen when the cells round out and lose contact with its neighbors. The nucleus fragment and the plasma membrane develop blister. The cells break into fragments then destroyed by white blood cell or by the neighboring cell. There are 4 stages of the mitosis. First is the prophase, second metaphase, third is the anaphase, lastly is the telophase. Mitosis basically involved in a period of nuclear division and also the cytoplasmic division. This diagram shows the early prophase during the beginning of the mitosis. We can see here that the duplicated chromosome begins to condense. During the late prophase, new microtubules are assembled. One centriole is moved toward opposite pole of the spindle and nuclear envelopes start to break up. And this is the transition to the metaphase where the spindle forms. Spindle microtubule become attached to the sister chromatids of each chromosome. And this is the phase of the metaphase. All chromosomes are lined up at the spindle equator and the chromosomes are maximally condensed. We look at the bipolar mitotic spindle, which consists of 2 distinct sets of the microtubule, which each set extends from one of the cell poles. 2 sets overlap at the spindle equator, which function to move chromosomes during the mitosis. For anaphase, with sister chromatids of each chromosome are pulled apart. Once separated, each chromatid is becoming a chromosome. Finally is the telophase, where the chromosomes start to decondense. 2 nuclear membranes start to form, one around each set of unduplicated chromosomes. As a result of the mitosis, two daughter nuclei has been produced, each of same chromosome number as a parent cell. Chromosome is in unduplicated form. For the cytoplasmic division, usually occur between late anaphase and also end of the telophase. It involves 2 mechanisms, which is cleavage in an animal and also cell plate formation for plants. This is the illustration for the animal cell division. So what happens when the cell lost its control? The growth and the reproduction depend on control over the cell division. For the checkpoint proteins, growth factor invite transcription of genes that help the body grow. Other proteins inhibit cell cycle changes, such as after chromosomal DNA get damaged. When all checkpoint mechanisms of a particular process fail, the cell loses control over its replication cycle, and this is how the cancer can happen. Neoplasm are abnormal masses of cells that are lost control over how they grow and divide. There are 2 types of neoplasm. For benign, is grow slowly and retain surface recognition protein that keep them in a home tissue, which is non-cancerous. And another neoplasm is a malignant, which grow and divide abnormally, disrupting surrounding tissue physically and metabolically, which is also known as a cancerous cell. HeLa cell is one type of the cancer cells. It is a line of human cancer cell that can be grown in a culture. Descendant of tumor cell from a woman named Henrietta Lacks. Lack died at 31, but her cell continued to live and divide in labs around the world. For this process, you need what we call as a culturing cell or normally known as a cell culture. Growing cell in culture allows researchers to investigate process and test treatment without danger to patient, and most cells cannot be grown in culture. Okay, time for break. We will continue later.